Dr. Yam, welcome and good to
Hi,
see you.
hello, good afternoon.
It you know uh, this is something that a lot of our faculty have already had a chance to do, and it's good that I finally got a chance to sit down and talk to you. And this is true. Almost all of our faculty they're coming from different areas of the world, a lot
Mhm.
of different backgrounds, a lot of education, a lot of high level education from some of the best universities around the world. And everybody's story is a little different. I love to hear things about what got you to get into the work that you're doing, and what inspired you. Um, Going through all of that training in your life, or periods in your life when you were making this decision, I want to get into science and understand some of the things related to what you can do to affect people who have corneal blindness.
Yes.
What led you, or what was the inspiration behind getting started in this type of field?
Okay, so this is something about related to my family. Yeah, so it's about my elder brother. So I just have one elder brother. So he is 3 years older than me. And actually he has glaucoma, or specifically primary open-angle glaucoma since very young. So I remember it's about 10 to 12 years old. When he was 10 to 12 years old. So so far, so throughout so many years, he has a lot of different treatments, including topical eye drops, Sanatan, Lumican, Timolo, and so on. This give him very hard life actually, is because there are a lot of side effects and so on. And also he at different period of time, he also have the laser trabeculectomy surgery to open the drainage
Wow.
to lower his intraocular pressure, and also he has several surgeries to implant the shunt device,
Mm-hmm.
and including the latest one is last year using the InFocus. But all this cannot control well on his intraocular pressure. So his eyesight dropped a lot throughout the years, and right now he's in Toronto, and his eyesight is only about 20 to 30% of normal vision.
Oh,
So he can read newspaper, he can work on the computer, he can read his phone, and all this have to rely on the voice assistants. So this gives him a very poor quality of life.
hmm.
So that's why when I graduated with my PhD degree in cancer cell biology in Hong Kong, I got a chance to interview in the eye department of Hong Kong, the Chinese University of Hong Kong, the ophthalmology and visual science department. So they look for researchers doing different type of researches, including the genomic research on glaucoma in Chinese subjects. So this project interested me, and that's why I started to do the eye research, because I'm afraid that at some point of my life I may also develop glaucoma.
Mm-hmm.
Yeah, so it's a very personal reason. Yeah.
Well,
So
that's inspiring. I mean,
yes.
that's that. When I said inspiration, you had a real inspiration to do that.
Yes. I have my own reason to start doing the eye research. Yeah. So this is how I started, and then actually after my first postdoctoral in Chinese University of Hong Kong, I went to Switzerland to have my second postdoc position, my second postdoc fellowship in U of Zurich under Prof. Jürgen Roth. And actually at that time, I had more intense research on glaucoma. I spent three
Okay.
years in studying one of the glaucoma causing gene, that is the myoxylin. Yeah, and I even showed that the mechanism, how this myoxylin, the mutation of myoxylin cause glaucoma. For example, I showed that some of the disease-causing mutations are actually misfolding the protein. They misfolded the protein, causing the protein to mistraffic inside the cells, causing the cells to undergo cell stress, leading to the death of the trabecular meshwork cells, and then loss of function in the aqueous humor drainage, increase the intraocular pressure, and then damage the optic nerve. So that can cause the onset of glaucoma. Yeah. So I spent 3 years in looking at that, and also I also work on some small molecule drugs to correct this protein misfolding. And this drug has been right now, because the result has been validated by different groups and studies. So this means that this drug is really improving the treatment of glaucoma. And right now, this drug is even used in different clinical trials of liver cholesterol dysfunction and also cystic fibrosis. So this is one of my achievements in the glaucoma research. Yeah. So it's, yeah, I'm quite satisfied with these results in Switzerland. And then after my postdoc fellowship in Switzerland, I returned back to Hong Kong as the assistant professor in the ophthalmology department again in the Chinese University of Hong Kong, and then started my work on the corneal stem cells. Okay. And also the corneal cell therapy to treat different types of corneal diseases. And that's why now I'm here in Pitt, working on the stromal cell regeneration therapy for corneal scarring.
Yeah, you know, the last part of your story, I knew. I didn't know all this stuff beforehand, which is really fascinating. And you know, the cornea stem cell course is, you know, something that the university, as I mentioned, has been working very hard at trying to solve the problem for cornea scarring. And we had here, you know, a world-renowned research scientist, Dr. James Funderburg, who sadly we lost last year, which was difficult for everybody. But in the process of all of that going on, you know, our leadership within the institution also found you and said, you know, this is someone who's doing work that can come right in and step in and take over the work that Jim was doing, which again is so very important. So without going into the details of that work myself, I wanted to have you describe what you know this process is in regenerating the cornea in people that have had scars that actually have left them with no vision, and you know, in I guess the most you know, plain language that you can provide for us. How does that work? What are we doing here?
So for this corneal stem cell treatment that Prof. James Funderburg has been developed for over 10 years, 15 years, it has been very good results showing by different basic and also preclinical trials using the animal models, because these stem cells are actually the progenitors or the precursors of the mature cell type in the corneal stroma. The mature cell type is the corneal stromal keratocytes that play a lot of important roles in maintaining the cornea to be strong, to be transparent, and also to have the refractive index strong enough that it can converge the light perfectly onto the retina. So, but then the corneal stromal keratocyte is difficult to reproduce or to generate because actually I have my past experience. I worked in Singapore for 7 years, and I have 7 years experience of doing this keratocyte research. And there has been a long-term problem that the keratocytes cannot be replicated or propagated in cell culture, because these cells are easily prone to form another different cell types, that is the fibroblast, that is involved in the scarring and also in the wound healing. So in my year in Singapore, we had developed a special protocol to generate these cells. So at that time, Jim and I are working, actually, we are kind of working in the same field, because he worked on the corneal stromal stem cells. I work on the mature stromal stem cells, that is the keratocytes. So we are meeting a lot of times, every time in the eye research meetings, conferences, and so on. So we have a lot of interactions and things like that. We have a lot of discussions. So I was also very amazed by his work on the corneal stromal stem cells, because these cells are more easy to propagate, to expand in number. Yeah, because they are stem cells. And also they can be, Jim also identified these cells in a particular region in the cornea. So we can easily isolate that region and get highly purified cells of these stem cells, and then generate this into a more cell number that can be used for different purposes, including the clinical applications, research, and so on. Yeah, so Jim did a very fantastic job. And I'm really sad when I heard the news that he passed away last year. And this is really a big loss to this department and also to the whole world of vision science. Yeah, So that's why when the colleagues here in Pitt asked me if I'm interested to continue with Jim's project, because this work is quite mature, that he showed very promising results in all the basic and also the animal models. And also there has been an ongoing clinical trial in India that they are using these stem cells for patient use. So when the Pitt colleagues they talked to me about this, and they asked, okay, if we can bring these techniques or this kind of treatment back to US, that would be wonderful. So since I have this background of doing the stromal cell therapy, so that's why I think, oh, why not? This is a challenge for me. And also, this actually is also close to the field of my research on the corneal stroma. And I have the basic knowledge, I have the basic techniques in doing the corneal stromal cell therapy. And I think I would be more easy to pick up all the things that Jim left behind and continue his work into a more, just to reproduce it and also to make it even further, to develop it into a clinical relevant model that can be applied in the patients.
Well, I know that that's certainly the plan, and you know, and you described it very well in terms of what you know, where we are, and you know, I know that the hurdles are going to be significant because we have to go through, you know, the process of getting these to be you know, manufactured in a way that you can provide them to patients, and it has to go through the FDA regulations related to all of that.
Policies, yes.
But when there's proof of concept that's already been done, and we're at a stage where we can do, you know, we know that we can have success once we get through all of those regulations.
Yes.
It's got to be pretty exciting, and I certainly am with you in being excited about the potential of what we can do for patients. Yeah. So, you know, thank you, and we're excited about that for sure.
Actually, right now, what we are doing with this project is to convert our lab-based protocol into a GMP, good manufacturing practice compliant protocol. That's to make sure that the cells that can generate for future patient use must be safe. That there's nothing that in contact with any animal origins, but their materials with animal the origins, and also they have their good efficiency in the treatment of the corneal scarring, to prevent corneal scarring or prevent corneal haze, and also to reduce the corneal scarring. Yeah, so this is what we are doing at the moment, is to adapt a GMP compliant conditions for the cell culture. And in the future, in the next phase, then we'll use these cells and test these cells in different animal models to make sure that these cells are good, safe, and they are working with high efficiencies to reduce the corneal scarring.
That's fantastic. So I you know, I'm not going to put you on the spot with questions like this, but what people would want to know is how long that could take. Any word for people on that in terms of how long we may be able to get there?
Of course, we are doing our best to speed up the project, especially in this difficult time with the pandemic and also the lockdown. We have locked down the lab for 3-4 months, and we are trying to get back the lost time from the lockdown. And that's why we are doing our in full gear, doing our work. And hopefully this stage of converting into a GMP compliant protocol won't take too long, especially if we have more materials to throw in, and also with good machines. But we still have to spend some time on this, is to make sure the results are repeatable. Mm-hmm. And also the results are truly reflecting the safety of the cells. Yeah. So this may take another maybe 2 months or so that we can 2 to 3 months. So I think by early next year, maybe we should have an idea of our GMP protocol, and then when we are still optimizing, we can already start the animal work to test these cells. Yeah. So all the steps will be overlapping. Yeah,
Okay.
so we are not waiting for the finish of one step and then move on to the other. So we are overlapping it and then trying to get back the lost time due to the pandemic.
Yeah, well, that's wonderful because we know there are a lot of people waiting. So that's wonderful to hear that that's the plan. With your coming to Pittsburgh and the decision you had to make. Obviously, you said what factored in is that you could follow up in a legacy of some really good work, and that's already a stage where we can make something happen, and that makes complete sense. But you have amazing background. You have a lot of work that you've already done, and I'm sure there are other places you could have gone, and to carry on this type of work. Um, with what's developing here with the department in terms of the breadth of knowledge base that's being developed here in the work that we're hoping to do in all these areas, is that at all a piece of where your appreciation, I guess, for Pittsburgh and coming to Pittsburgh may have been part of that equation, or what does all of this mean to you coming to
why I come to Pittsburgh?
Yeah.
Okay, so now I think after my PhD degree, after I got my PhD degree, I have quite a number of years, over 10, 12 years of doing ophthalmology in different places, in Hong Kong, in Singapore especially. My last 7 years was in Singapore from 2012 to 2019. And I work with, actually it's a very good cornea surgeon and a professor. He's called George Matter. He's a very nice physician, very knowledgeable, very productive. And the most important thing that I learned from him is that we have to make our research translatable. So we cannot just stay in the research lab and then do our own for our own interest, but we have to apply our work and our research to the patients. So this is the bench to the bedside. And also we have to understand what the clinics needs and their questions so that we can do something in the lab to solve this problem. So this is putting the bedside back to the bench. And this requires very good interaction and communication between the lab and the clinics. And this is actually what is happening here in Pitt Ophthalmology under the leadership of Dr. Sahel. There's a very close interaction, very close connection between the physician and the scientists. And I actually appreciate this opportunity so that we can build up further on this relationship to make it more strong and more productive. So that's why this is the reason why I choose to come to Pittsburgh, because I know that here we have a strong background of doing translational research. And this is what I learned in my so many years of ophthalmology research.
Yeah, that's certainly encouraging and refreshing to hear, because I think it's very true. I think that Dr. Sahel's model is essentially just that, looking towards the patient's needs, and building the team to help us develop what's needed to be able to help that patient. And you're part of a whole task force of people that are working specifically on that. That includes physicians, surgeons, Dr. Dollywall and Dr. John G, I know, and then you work with other scientists as that team, and you work really, really well together. I've been able to attend some of those meetings with you, and I think you work really, really well together.
Yeah, we have a close tie together, and we just work like in a family, that we just help out each other in doing different type of, because no one can do everything. Mm-hmm. And we need, especially we are doing medical research. So as a basic scientist, we know most of the things in the lab, but we may not have too many knowledge about the clinics, and we don't know what the clinics needs. So that's why we need to have a very close interaction with different clinicians, and often talk to them, discuss with them, and then see what we can do from the lab that can help them to achieve better patient care or the patient treatment.
So this is a wonderful story for the, from where you started in getting interested in science and, wanting to do things for people like your brother who are dealing with glaucoma, to where you're, leading something that can be very important for not just the Pittsburgh and the university in Western Pennsylvania, but for the world, developing safe practices to develop a way to clear clouded corneas using a stem cell technique, which was developed here. And I think, you know, it is a good position, and I think that we can accomplish that goal. And again, we're proud as the foundation to be able to help support that. What will be next, Gary? What will be the next, frontier when we tackle this? Because I think we're gonna tackle this. What would be the things you want to see for the future that you would really like to focus on, and that you feel that you can contribute in terms of this type of because you're still a young guy and you have a long way to go.
Yeah, okay. So, so my immediate target is on this stromal cell-based therapy to make it GMP compliant or approved by the FDA, that we can launch clinical trial to test on patients so that we can get preliminary data on how the patient's treatment outcome is and so on. So this is my immediate task, okay? But this is still involving some kind of cell therapy, cell surgery, and so on. We have to put the cells there. So my next one, my another aim is to look at some kind of so-called cell-free therapy for the cornea scarring or the opacities or corneal clouding. Yeah, because the cells, when they are put on the corneal surface, actually it may not be the cells that can help, that can do the main job, but it's the secretion from the cells. Something that expressed in the cells, some molecules, some proteins that express in the cells and being secreted out to the cornea tissue that can help the other resident cells to survive better, to do their job, and so on. So this is what I'm looking at, is some soluble factors, some molecules that can be secreted from the cells. And then hopefully we can identify some of these molecules and then develop it into some kind of eye drops or some kind of very feasible cream, eye gels, and so on, that we can apply onto the patient's eyes instead of doing surgery. So
That's
this
fantastic.
so this is something that we are working along this direction. And then the other work that I'm interested in is it's not on the corneal stroma, it's on the corneal endothelium regeneration. Yeah, because corneal endothelium failure is a major indication of corneal transplant. More than 70% of corneal transplant is to replace the defective corneal endothelium. So right now, my lab is also working very actively to look at cornea's endothelial stem cells. Yeah, so we have a paper last year, and we are going to have the second paper coming up, is to use these corneal endothelial stem cells to generate functional corneal endothelium that can be used in animal models of corneal endothelial failure, helping the cornea to get clear, to get back the clarity, and also to reduce the corneal edema. So if this approach is feasible, is correct, so this will be a future treatment modality for corneal endothelial dysfunction or diseases like for the Fuchs dystrophy.
Wow, that's amazing. You know, congratulations. There's a lot going on.
Yeah, we are.
But it's amazing that you're able to keep your focus on so many things simultaneously. It really is. We're very fortunate to have you here in Pittsburgh, and I'm glad that you are part of this team. So thank you for coming and sharing this with others as we again are working towards these goals, and it's important to let people know what's needed, but also what the future plans will be. And I'm really encouraged after listening to you today that I think that we are on the right track and we have the right people doing the work to get that done. So thank you.
Thank you very much. Yeah.
All right. You have a good rest of the day.
Okay. Thank you very much.